you ask me what is the dream, what I'm interested in is not only doing knockouts, but it's doing knock-ins.
Yes.
If you want to do knock-ins, you need first to have an efficient cut. So that we can achieve in pretty much every type of cells, right? But then you need to deliver a donor template that will be used to repair the cut and integrate the piece of DNA you're interested in. And then you need also to have cells that are dividing, and it's better if it's have a synchronized way of dividing, because the homologous recombinations that is necessary to repair the DNA and integrate your gene of interest only happen in dividing cells. And fast dividing cells also tends to do more. So here I think, my dream is to be able to do knock-ins in every type of cells. And right now we can do very efficient knock-ins in T cells. We start being able to do good knock-ins also in NK cells. But yeah, the big issues is delivering the donor template. So we are using, for example, adeno-associated viruses, which is a good way to protect your DNA when it goes through the cell, because DNA in the cell is toxic. It's recognizing as an infection or damage. So it's protecting the DNA until it's released in the nucleus. But there are other methods that people are using. Naked DNA associated with a lot of toxicity, though. But if you work with T cells that grow so much, you can maybe deal with that. But then, being able to also deliver the CRISPR or other gene editing tools into the cells that are sensitive, like NK cells, for example, are much more sensitive. They tend to die when you electroporate them more, or they stop growing when you electroporate them. So, my dream is to be able to do all these modifications without losing 50% of the cells.
Yeah.
And being able to make them divide enough so you can achieve some knock-ins. And I think there's still a lot of space to improve.